Hello and welcome back to today's video. It is part 2 of the chapter 9 series and it is on gene regulation, following up on the gene synthesis, protein synthesis video. Let's get into it. Here is our learning goals from the QCAA. 1. To recognize the purpose of gene expression. It is to synthesize a functional gene product, and this process occurs and is used by all known life. There are a few factors that regulate gene expression, and they occur at different stages: transcription, translation, and due to environmental exposure. We will also look at how these transcription factors affect the formation of tissue and morphology, and the Hox transcription factor family has a big part to do with that. So you can pause the video here and summarize those key points. Let's get into it then. This is from 9.2 and 9.3 of the textbook, and it's on gene regulation. So we know that every cell in our body contains the same genetic material. So how do our eye cells know to become eye cells and our stomach cells to be stomach cells? We want our genes to be regulated, controlled. So DNA needs to be able to be read and particular sections to be turned on and turned off. That is what gene regulation is. In eukaryotes, such as you and I, we have enzymes and factors that allow sections of DNA to be transcribed and translated. Prokaryotes, such as bacteria, have another structure and method that allows them to regulate gene expression. It is called the Operon. This particular structure is found predominantly in prokaryotes, and it allows the body, the cell, to tell which genes it should express or not, and when. Let's look at E. coli, for example. They are a bacteria that live within our intestines, and they consume whatever we consume. So if we have an apple, they must break down that apple to obtain energy. If we have a glass of milk, then they need to be able to break down that lactose to obtain energy. There's a few parts to the Operon, this feature that allows it to gene regulate. I'll pull up this diagram now to talk through these parts. So here, imagine we have a double strand of DNA. On that DNA, there are different regions made up of nucleotides that code for genes. So here we have a region coding for the LAC-A gene, the LAC-Y gene, the LAC-Z gene. But further up the DNA stream, we have this section called the Operon, and it's made up of a few parts. The first part that I will talk about is called the promoter. It is like the primer that allowed DNA polymerase to know where to begin replicating DNA, if you think back to that lesson. The promoter similarly tells RNA polymerase where to begin, where to start writing that mRNA message to then in turn create that gene, that protein, to express that gene by creating a protein, I should say. It's a sequence of genetic material, DNA, that RNA polymerase can physically bind to begin making that mRNA message, which will eventually be used to make that protein. That's the promoter. The next section we have here is called the operator. The operator is part of the DNA where something called the repressor can bind to. The repressor, if bound to the operator, blocks RNA polymerase, blocking mRNA, therefore, from being written, and thus these proteins downstream from being expressed expressed, from being formed. So let's look at an example of this in prokaryotes by talking about this lac operon. It's called that because it's in reference to lactose. There are 3 genes that we can see here. These 3 genes code for enzymes that help break down lactose. Why would bacteria need to break down lactose? Well, remember, they're living inside of us, and what we drink, they drink. Lactose is a sugar that bacteria will want to break down so that they can metabolize it, so that they can continue living and multiplying. The lac operon, why are we talking about this feature? Well, it's this structure that allows bacteria to gene regulate, and it's the first one that we kind of came to understand. It has another gene that codes, and so this section up here, there is this section that codes for the production of the repressor. And so let's talk about how the lac operon works now. If lactose is present, so after we've consumed a glass of milk, then this lactose, let's talk about this section, it binds to the repressor. And what happens there is that the repressor physically begins to change shape, therefore, and thus releasing it from the operator. Now that the repressor has been released from the operator, RNA polymerase can then come in and bind to the promoter, thus creating that message RNA, which will then be used in the production of the enzymes and that expression of those enzymes that are used to break down that lactose. So when lactose is present, it binds to the repressor, changes its shape. RNA polymerase can come in and start working and now writing that message to create those enzymes. So this happens really quickly when we drink a glass of milk. If we talked about now, what happens when no lactose is present? Well, in the absence of lactose, the repressor remains bound to the operator, therefore inhibiting RNA polymerase from coming in and expressing those genes. So why is that important? Well, it's about conserving energy and materials. We wouldn't want to be creating all these enzymes if we didn't need to. That's a waste of time, and it's a waste of resources. So this whole process is an example of how bacteria can gene regulate. It's known as positive control because if lactose is present, then it produces these enzymes. I'm going to talk about another example of an operon in bacteria that is the opposite to this, negative control. And it is called the tripoperon. So the tripoperon, an example of negative control, is actually what we can say off when tryptophan, the product that it's in reference to, is present. And instead of having 3 genes here, there are 5 genes. So the tripoperon, instead of the lac operon, encodes for genes that will allow for the synthesis, the production of this substance, tryptophan, and that's really important. It is regulated by the amount of tryptophan already present. And tryptophan, instead of being in positive control, it actually acts as a repressor. So let's talk about that now. When tryptophan is present, it actually activates the repressor and stops RNA polymerase from working. When tryptophan is present, it binds to the repressor and it activates it and stops RNA polymerase from coming in and writing that message to produce these enzymes, which will then go on to produce this substance called tryptophan. So again, why is that necessary. Well, we don't want to be producing these enzymes to produce tryptophan if tryptophan is already present. So this is an example of negative control. But in the absence of tryptophan, the repressor dissociates, it comes away from the operator, and therefore RNA synthesis proceeds. RNA polymerase can come in, read the message, produce these enzymes, and therefore start to produce tryptophan. Tryptophan is essential in producing like hormones that are necessary in eukaryotes for good skin, for good sleep, and also producing serotonin, what we call the love hormone. But we don't have this Operon. We actually have to obtain tryptophan from our diet, and turkey is a really good source. But how's this? Little bacteria, simple prokaryotes, have this feature called the Operon that allows them to turn on this ability and turn off the ability to produce tryptophan when needed. Just like it can produce these enzymes that break down lactose when lactose is present or not. So that is the Operon. These two examples of gene regulation are occurring at the transcription stage. That is the stage where that message is being written. So if it came after the transcription phase, then we could say gene regulation is occurring at the translational stage. I will talk to that more in a moment now. If you need to pause the video and go back over what I just spoke about, then you can do that here. Okay, let's move on. So, to help understand this section, I want you guys to create a table with me. So grab out your ruler, and it doesn't have to be a huge area, but maybe about half a page worth. I'll show you how to set that table up now. Let's create this section where we have what is the regulator. So is it lactose that's going to regulate whether those enzymes are produced or not, or is it tryptophan? What type of regulator is it? There's another column. When does it work? And how does it work? So if we spoke about those two examples that we just did, let's put that in. Tryptophan and lactose. They are our regulators. If we spoke about lactose, for example, what type of regulator is it? Well, those enzymes are produced, those proteins, based on whether lactose was present or not. That's what we can call an environmental factor. When does it work? Was it happening when the message was being written to produce those enzymes, being the transcriptional stage, or was it coming after that, being translational stage? And how does it work? So if we fill those columns in for each of these regulators, then yeah, you're preparing yourself pretty well to meet the criteria, I feel, at the start of this video. Pause the video and give it a shot. Okay, welcome back. Let's go through it together. Tryptophan is an environmental factor. We know it works at transcription stage, and it essentially stops the production of tryptophan. So when it's present, it doesn't need those enzymes to produce it. Lactose, on the other hand, is an environmental factor, again, transcription, but it activates the production of enzymes to break down lactose. So positive example of gene control and negative example of gene control. So you can pause the video here, fill those in. There's a few more rows that we're going to fill out as this video continues. Okay, so 9.2. That's what happens in bacterial cells, in prokaryotes. What happens in you and I, or in animals, in eukaryotic gene regulation? We don't have operons. So we have transcription and translational factors that control gene expression. I'm not sure if I mentioned it, so I might have, but I'll mention it again. By the way, we still need to obtain tryptophan for our body, for good sleep and good skin and all that. turkey is actually a great source of tryptophan, and we obtain it largely from our diet, by the way. Okay, so yeah, we don't have operons. We have what are called transcription and translational factors that control gene expression. We don't want our eyes to start producing what our stomach cells do, right? Hydrochloric acid, that would burn. Watch the Amoeba Sisters video, it's pretty funny. Let's bring up this image here. If you looked at that, I hope you would be able to identify. Essentially, there's this histone complex, these 8 proteins, and DNA is wrapped around it, and that is called a nucleosome. What we haven't seen in the other diagrams that we've looked at is that these nucleosomes actually have these tails coming off the side of them. And those tails are really important in the process of gene regulation for us eukaryotes. We can do things to these tails to control the expression of DNA, which genes should be turned on or off, depending on what cell it is. The first thing that I will talk about is called acetylation. And when you acetylate the tails, it means you add an acetyl group, a chemical compound of acetyl to those tails. This then causes an effect through charge on the histones, causing these nucleosomes to repel each other. That unwinds the DNA between them, and it physically allows RNA polymerase to come in, to attach, and to begin writing a message to translate that gene into a physical protein. Methylation is the opposite in terms of when a methyl group is added to these tails through, again, talking about charge. We don't need to go into the chemistry of it. It actually causes these histone groups to come closer together, therefore physically blocking RNA polymerase from coming in, therefore prohibiting transcription. So if we added that to our table now, acetylation. It's a process affecting the histones or the DNA. It again happens in transcription because we're talking about RNA polymerase. And it allows transcription, unwinds the histone sections. Whereas methylation or methylation, again in transcription, it prevents transcription from happening because it brings them closer together. RNA polymerase cannot come in. So as the name suggests, if you have, and by the way, methylation is a process that happens in eukaryotes to actually determine fur color. So, gene regulation. As the name suggests, if you have a gene regulatory protein that gets involved, it interferes with the transcription process. So it's simple as that. So a gene regulatory protein interferes with this process. So here's products of other genes. It again happens in transcription stage, and we don't have to go into more detail than that. The QCAA learning goals simply said to identify and to recall. So if I keep moving on then, mRNA binding proteins. I'm going to take that back. If you know that gene regulatory proteins inhibit transcription from taking place, then what would mRNA proteins do? When is mRNA used to do something? Is it in transcription or translation? Well, we know that mRNA will be acted upon by a ribosome to produce a physical protein. So, a mRNA binding protein stops this process of translation from occurring, therefore preventing that gene from being expressed. Lastly, we have what are called microRNAs, and they are, as the name suggests, just these really short segments of RNA, no more than 20 base pairs. And these microRNAs bind to and stop the ribosome from doing its thing. So we can add that to the table here. Prevents, again, translation. Ribosomes cannot read that message RNA because of what are called microRNA. So these are a few things that eukaryotes do to control what genes are expressed or not. So you can pause the video here and fill that table in. I'll talk to this table a little bit more on the next slide. So thinking back to the QCAA, and in terms of the final exam now, what's the goal of going through this? Well, I hope that you would be able to read a question, and maybe it's talking about tryptophan or the trip operon, and be able to remember, oh yeah, that was an environmental factor. It worked in the transcription stage. And if you can even remember what it does, then maybe a question where it says describe what happens, that would be able to help. But the takeaway message from this table is this: to know that there are different factors that work in the transcription stage and the translation stage of gene expression that can either stop a gene from being produced, protein from being produced through that gene being expressed, or to allow that to happen. And the more that you can take away from this, the more that you prepare yourself for an exam that might ask you to describe that process. And I hope that that makes sense. Check out the description of this video for some, again, awesome videos that just talk about this in another way and provide different visuals to help you understand if you need it. Then you're also reading the textbook, so you're covering a few bases there. Okay, a couple of questions quickly. Identify which of the following is a factor that regulates phenotypic expression of genes during translation. That's the kind of question that you might be asked. This is from QCAA. So pause the video, give it a shot, then we'll talk through it. Okay. Well, we know that the repressor and the promoter are parts of the operon. We haven't talked about the activator. We have spoken about microRNA, and that is a translation factor. It prevents translation. So the answer is D. Question 11. And by the way, QCAA says to identify and recall. So those are the verbs coming in play here. It's not asking you to write a big short answer on microRNA. Don't need to do that. Question 11. A group of genes that control the pattern of body formation in humans, morphology, so that's like thorax, abdomen, is being investigated. A gene would be part of what group? Pause the video here if you need to. We haven't spoken about this, by the way, but it's in your textbook readings. From 9.3, and it just mentions that Hox genes are the ones responsible for, in insects, saying there's a head, there's a thorax, there's an abdomen, there's a tail, that kind of thing. So the answer is A. And that is all for this video. Again, from the Amoeba sisters, I think humor can be a helpful way to help us learn things in a different way. So this one is just talking about transfer RNAs carrying amino acids, and Ted and Terry, the transfer RNAs, didn't mind the job, but some of the amino acids were a real pain. Guys, I hope that this video has been informative, and I will see you in class. God bless.